So I'm going to talk first then about the question of understanding antibody responses to vaccination. And this bridges chapters 1 and 3 of the Post Vaccination Monitoring Guidelines, because we use the responses to vaccination both to help us select vaccines, which is chapter 1, and also to help us look at how good a job we're doing with the vaccination and whether we've managed to immunize the population at large in chapter 3. So what I'm talking about here are the antibodies to the foot and mouth disease virus structural proteins, which we abbreviate as SP antibodies, and how they can be measured, and how the findings can be interpreted. And I'm not talking here about NSP antibodies, which we use for DIVA serology to detect infection in vaccinated animals or infection generally, and that topic will be dealt with later when we come to look at how we can monitor the success of vaccination programs. One thing I would say about this is some of these concepts are a little bit complicated, and especially for non-lab people, and that's because of the problem that the correlation between protection and antibody responses is not all that straightforward, and so that does mean that it's a little bit complicated to try to explain what's going on, but I hope at the end we can still see that there are some practical and reasonably straightforward things that you can do and have confidence in. But anyway, I'll try to work through the issue of these correlates of protection in the next few slides. So antibodies to the FMDV structural proteins, these are the ones that are serotype specific, that's how the serotypes are defined, and they correlate with protection. And you can use, the laboratory can do a number of different tests that measure these types of antibody, and common ones are the virus neutralization test using cell cultures and live virus, or various SP ELISA tests, such as the traditional liquid and solid phase tests and commercial alternatives to the solid phase tests that are now available. And according to how you configure these tests, you can measure the amount of antibody either specifically to the vaccine strain, which is therefore the homologous response because it's the response to the same strain as you've been immunized with. You can measure to the field strain if you include field virus or field antigens in your test. So that is the heterologous response because it's not the same virus as was in the vaccine strain. Or in your test you can incorporate a reference strain of the required serotype. And that will give you, if you like, a general response from that serotype, but not specific to any one particular strain. So if we look at this correlation between SP antibodies and protection, the fact that it occurs is useful, as I've sort of indicated, for the vaccine matching, for selecting the vaccines, and also for checking the quality of vaccines for purchase and later on for post-vaccination monitoring. But the minimum amount of antibody for protection, the threshold that defines protection, is very difficult to define precisely, and that's because it differs according to quite a number of different factors, and this is where it starts to get a little bit complicated. So for example, different animals seem to require different levels of antibody in order to be protected, even where you're using the same viruses, the same tests and all the rest. There's animal to animal variation, and that's probably because maybe the tests don't measure the correct type of antibody, or maybe the antibody tests aren't measuring other forms of immunity, such as cell-mediated immunity, which can also contribute to protection. A second problem, which we've sort of touched upon, is to do with the viruses, that different viruses actually also differ in the amount of antibody you need to protect against them. So that's also means that to some extent the results are specific to different vaccines. And as we say, the tests and which reagents you include in them, whether you include the homologous reagents or the heterologous reagents will give you a different result because there are antigenic differences between the viruses. And also for some tests, such as the virus neutralization test, the test is itself not that reproducible between different iterations of the same test. So all of this can lead to variability and a difficulty in interpretation. And finally, the time after vaccination can also affect the correlation between antibody level and protection. So if you look 3 weeks after vaccination, there may be a different level of antibody associated with protection than if you look 5 months later. So what are we going to do about all this? And that all sounds rather discouraging, and maybe we would think it's not possible to do anything with antibodies, but we still go back to the fact that there is certainly some correlation between antibodies and protection, and it's the best tool we have to measure the immunity to the vaccine. So what can we do? Well, the gold standard method is to establish the correlation from a potency test with challenge. And I talked about how you do a challenge test in the first webinar that I did last week. So as we described, you can vaccinate animals and then challenge them from 21 to 28 days later. And most of these challenges are done with the homologous virus, and they're done as a part of the regulatory requirements for showing that the vaccine is effective. So it's a standard challenge procedure. And you can collect the sera at the time of challenge, and then you can correlate the protection in each animal with their serology results. And hopefully you find that there is a correlation between antibody level and protection. And if you find that there is a correlation, then you can use serological tests in future batch release potency indicators. So when you come to make more batches of the vaccine, once you've shown the correlation with serology, you can do a vaccination test without challenge, and then you can show that the vaccine has induced the required level of immunity. And then finally, an important point, that if the batch release sera from these vaccinated animals are supplied with the vaccine, then these can be used to calibrate your own tests. So these are very useful things to ask for in your tender specification, and I was pleased to see that quite a number of people had picked up on this and asked for this in their tenders. So what follows now is a couple of tables that show where people have looked at what is the actual correlation between antibody thresholds in different tests with vaccine potency tests with challenge. So this first slide shows a paper that was published some years ago, and they were looking at 3 serotypes, OA and Asia 1, which, if I can find the arrow, yes, you can see here on the left-hand side of the table. So we've got serotype OA and Asia 1, and then we've got along here, these are the antibody titers that are associated with a 50% probability of protection or 95% probability of protection, and this is the confidence interval for the 50% protection. So for example, if you go to serotype O, you can see that a titer in the VN test, a reciprocal titer of 40, was associated with 50% protection, but that the range was quite wide between some animals were protected with as low as 13 and some needed 50. And you can see that there are different values for each of the different serotypes, and for some of them the confidence intervals are quite wide. So if we look at what is the most common requirement for a vaccine, which would be 3PD50, a 3PD50 vaccine equivalent is equivalent to a 75% probability of protection. And if we look at the average data from these 3 serotypes, we get that the titer on average that will give you 75% protection is around 1 in 56 in a virus neutralization test. So that's how you can come up with a sort of general figure for what on average is associated with protection. And this second slide shows similar data from South America for using the liquid phase blocking ELISA. And again, you've got some different viruses or vaccine strains on the left, and you've got differences in antibody titer that are associated with 75% protection. So ranging from 1 in 79 all the way up to 1 in 158. So again, you can see it's not always the same for different strains. So it says here at the bottom, titers are specific for different vaccine strains, and titers may be specific to the time after vaccination, which we have already mentioned. So in real life, there is a tendency to use these rule of thumb approximations rather than properly established cutoffs. And this is where we get these figures of around 1 in 50 for VNT, which are assumed to be adequate, and around 1 in 100 for the liquid phase blocking ELISA. But it's very important to remember that these are average values, and individual animals may not be very well predicted by these specific values. Also in real life, of course, it's important to remember that vaccinated animals are generally not challenged with homologous viruses, like in a potency test. In the field, they come up against viruses that are different to the vaccine strains. So they are heterologous viruses. And this, of course, can have a significant impact on the correlation with protection. So here we have some real data. These are actually from a study in Turkey, which Naji was involved in collecting the data. I know he's here doing the course. So you've got in this panel here at the bottom left, let me get my pointer again. Okay, so these are the titers. This is in the log scale. So 1 in 50 is about here, where this dotted line is. This is on the left-hand box, we can see the titers that are found in a group of animals that have been vaccinated, and then the test is done with the homologous virus. So we're looking at homologous antibody, and we've got animals that have been given 1 shot of vaccination and animals that have been given 2 shots of vaccination a month apart. And then we've waited until 6 months after this vaccination to see how much residual antibody was left. And you can see that for the homologous virus results, the animals that have had the booster are mostly up and around or above the level required to predict protection at the time of testing. But if you look at a heterologous virus in the test, which is similar to what is likely to be the virus that will be the challenge virus in the environment, then the situation is much different. So after all that, what is the actual simple solutions for establishing your PVM serology thresholds? So here are a range of possibilities. First of all, you can look at the expected response to vaccination and not worry so much about whether it's actually protective or not. So if, for example, you know that animals vaccinated with this particular vaccine under control conditions produce a certain antibody response, whether or not it's protected, that you can go and look to see whether you get that response in the field after you have vaccinated your animals. So this is a very practical way to proceed. You make sure you get a good vaccine that will give you protection, and then you look for the expected response in animals. And for this you need standard sera produced from animals given the relevant vaccine batch, either obtained from the manufacturer, which is why these batch release sera are useful, or if you can't get hold of them that way, you can produce them yourself in a controlled trial. A slightly better refinement is if there has been a homologous protection study done, you can then correlate the serology results with the potency test results, and you can assess whether the animals that have been vaccinated are likely to be protected against a homologous challenge. In terms of use in the field, this is only reliable as an indicator of protection if the vaccine and field strains are well matched. But as long as you know that that's true, then you're okay looking for homologous protection levels. If you think that's not likely to be true, then you really need to think about heterologous protection and correlating your serology with either potency results for heterologous protections, if known, but they're not usually known, or else you have to substitute field virus for vaccine virus in your serology test and try to estimate heterologous protection from that. And because this is quite a complicated and specialist field, as I expect you're beginning to realize now, it's very useful to work closely with the vaccine manufacturer and a reference laboratory who is familiar with performing these studies to help you establish the right thresholds. So I've mentioned that if you cannot get hold of batch release sera from the manufacturer, or if you have doubts in fact about the quality of the vaccine that is being offered to you, then it's quite useful to do a small trial yourself with some of the vaccine, maybe ask for a trial batch, or else do the trial before you go on to do a large-scale vaccination. And the objectives of this are to establish the expected titers for subsequent PVM serosurveillance, to check that the vaccine elicits an adequate antibody response to circulating viruses, to compare antibody titers between different vaccines, so you can see which ones from different companies and batches give you the best titers. And if you do a more sophisticated study and follow the animals over time, you can establish the need and timing of boosters, and even, if you want to, look at species differences between target hosts. So the methodology for this is to vaccinate a small group of animals of the target species. It's important to select animals without pre-existing immunity, and you need to keep the animals free from infection during the trial with some unvaccinated controls, which will show if infection gets into your trial. And you collect samples for sera at relevant time points, and you ensure that a sufficient volume is taken and archived. And these give you a very useful resource for monitoring your vaccine down the line. So to summarize all this, ideally you start off with a good match of the vaccine to the field strain has already been shown, and the vaccine producer can supply you with sera from batch release testing. So those, of course, are two key questions that you ask for in your tender: evidence that there is a good match and asking for these sera. If you have doubts about this, or if this cannot be fulfilled, then you can produce your own sera from a vaccination trial. You can test these in-house, e.g. with a commercial ELISA, and that can give you a threshold to use in population testing to see if your vaccination has been done correctly. You can also send these sera to a reference laboratory who is experienced in looking at protection levels, and they may be able to give you an estimate of suitability against specific field strains that they have in their collection. And they may give you an estimate of threshold to use in population testing to see if animals are protected.